I appreciate the opportunity to talk to the committee. I'm going to give a general overview just to talk about some of the organisms and some of the issues that come up with use of these gene drive systems. Okay, so I'll start off with the first slide here. Comes from the Ken Esvelt's article in 2014, which presented a very nice graphic about the kinds of organisms and kinds of issues that could be addressed by these gene drives. And so I'm going to go through, in a sense, this whole picture. And can you see my pointer here as I'm going around it? So I'm going to start off on one part of this slide and move through the different possible applications and give you a little bit more detail on each one of these. So I'll start by this with this little red thing circle around the specific area that I'm going to talk about. So first, aiding threatened species. Okay, what can be done here? And I'll give two examples, one where it seems to be a very useful approach, and another where it may not be as useful. So the first one I want to talk about as an example is the use of gene drives in terms of the Hawaiian honey creepers. Okay, so on the island of Hawaii, there's a group of birds that have diversified in the same way that Darwin's finches diversified, and they're beautiful birds, but they're endangered based on a number of impacts, some of them just environmental, some of them coming from invasive birds that come from the mainland that compete with them. And some of this is moderated by the fact that they are very susceptible to bird malaria. They evolved on an island where there weren't mosquitoes and there wasn't bird malaria, whereas on the mainland there was bird malaria, and species from mainlands typically have resistance that's evolved to this malaria. So about 100 years ago, the mosquito Culex quinquefasciatus arrived in Hawaii, and not long after that, bird malaria arrived. All right, so this is causing losses in the Hawaiian honey creepers, and there really is no other fix for this. And the birds are being forced up to the tops of the mountain ranges in Hawaii, and with global climate change, it's thought that their range will even be smaller. This is higher elevations have fewer mosquitoes and less bird malaria, as well as less competing species. So the goal of some researchers is to engineer this Culex mosquito that transmits the plasmodium so that we'll no longer be able to transmit that plasmodium, or to engineer it to decrease its populations. So that is something that's ongoing and could be quite useful. This mosquito also transmits West Nile virus, so there's other potential here in limiting its populations or stopping it from transmitting West Nile virus as well. So the resources may be different. Okay, but to look at a different situation, and in Kevin's presentation, he shows an amphibian here, and that's because there's a chytrid fungus that's causing massive die-outs of amphibians, and this is seen as a real problem, and sometimes the idea is that you could use gene drives to limit that problem. But that's a little bit problematic because you can't really use it to engineer the fungus, and the idea is to engineer the animals. But when you think about this problem, there's such diversity of amphibians. I'm just showing you just a picture to give you an impression of this. They're diverse, they're beautiful, but even just this group of glass frogs, right? There are 100 species, and you can't really rear them. They're hard to maintain. It would be very difficult to think about how you would approach this using gene drive. All right, so to move on, moving down here to looking at sustainable pest management. I'm not going to deal with rodents because that's something that John Godwin will deal with later on in the presentation. But there are lots of other pest situations outside of agriculture that one could address using gene drive. And one that comes to mind are roaches. The idea would be here specifically to use gene drive to limit populations. One of the problems, though, that faces researchers who would be interested in this is the fact that roaches actually produce egg cases. So if you look at, roaches don't produce individual eggs, but the eggs are enclosed in these cases. And one of the approaches that are being used today to do transformation, genetic engineering of insects, is to inject eggs. And when you have an egg case like this, it's impossible to do those injections. The needle's supposed to be very specific, and just cutting that away hasn't worked. But new technology is coming along that may enable researchers to inject females and have the genes go straight to the ovaries. And if that happens, then this is a possibility, all right? But I just want to give an example where it might not be as useful, and that's with bed bugs. Again, another major problem where we want pest management. But bed bugs, unlike cockroaches, don't move as much and typically establish based on one or a few individuals invading an apartment house or something like that. So the structure of their populations is very specific, and their mating is well known in terms of this is a male mating with a female where they just puncture a hole in the female and inject the sperm that way. So they have a very complex reproductive system that is very specific. So you'd have to be releasing the engineered bed bugs into each apartment complex or house, which probably wouldn't be that easy or even acceptable. With roaches, if you're talking about farms or anything else, a release would be more feasible. But I just want to give you a sense of how it's very important to understand the ecology and the genetics of the species to know whether these approaches would be valuable. And one thing that people talk about a lot is the idea that if you release these gene drives, you would cause eradication of an entire species, and we have to deal with the ethical issues of causing eradication of entire species. And this is a figure from the seminal article by Austin Burt in 2003 about using homing endonucleases that would decrease fitness. And if you look at this, you'll see that there's an equilibrium that comes about where the homing endonuclease comes to about a frequency of about 90%, and the population fitness goes down to about 20%. So if you think back about these roaches and bed bugs or other insects, their reproductive rates are quite high. So this would probably turn out to be management as opposed to eradication. Now, Austin Burt and others have shown that you could increase the utility of these approaches to cause this population fitness to go down even further. But I just want to point out that we're not talking always about a release that's going to cause extinction. Management may be what you get from some of these strategies. So to move on to agriculture, there are, again, pests where this technology is active in terms of research. One example here is spotted wing Drosophila, which is a new invasive pest in the U.S., across the entire U.S., causing lots of damage to small fruits. And this Drosophila is very closely related to the model organism Drosophila melanogaster. So doing the engineering is pretty much a very feasible thing to do. They have very large populations in some places, so the only way to really address them is with the gene drive. It's not clear how well this would work with this species, but given that it's a model organism and easy enough to manipulate, researchers are working in this area. But to look at some other agricultural pests, just to give you the extreme opposite, if you look at the coffee berry borer, which is a very important pest in coffee growing regions, people have been thinking about using gene drive, but coffee berry borer has got a very interesting biology where the larvae develop in the coffee berry as a group of related larvae, and when they become adults, they mate within that coffee berry. So there's no chance of releasing something and having it spread within the population. And another example I just want to point out are aphids, and this is an aphid giving rise to an offspring. No eggs are produced here, and they're typically asexual. So whenever you're dealing with something that's usually asexual, you can't really use a gene drive system that effectively. So in this figure, another example would be the western corn rootworm, which BT crops are being used for minimizing their numbers, but resistance is evolving to those crops, so people are considering the possibility of using gene drive to limit the population size of the western corn rootworm. But you have to consider the fact that there are, I'd probably say, trillions of western corn rootworms, and you have to think about how are you going to rear these. This is not an easy insect to rear to release the numbers that you would need to release. So these are, I'm just wanting to try to point out some of the ecological issues that determine whether a species is a good candidate for this kind of an approach. So to move on in terms of agriculture, so far we've been considering animal pests, but Kevin also brings up the possibility of using this for weeds and such. So there are some weeds where the use of gene drive could be interesting. This is just a picture of Scotch broom, where there's potential for use of gene drive. But I guess what I want to point out here about plants that's very important is most plants, the male and the female are on is the same plant, right? It's hermaphroditic, and many of the plants are selfing, so they self-fertilize, and that becomes the same problem as you have with the coffee berry borer, that you may release plenty of pollen or something, but if they're self-fertilizing, you don't get anywhere. And this just gives you a distribution of outcrossing rates. And even in cases where you have a high rate of outcrossing, you're really going to select those populations to become more inbreeding. And so you have to be very careful in terms of choice of plants. So to give you some examples, Palmer amaranth, which is very well known these days because it's become resistant to glyphosate herbicides. This is one of the rarer plants that actually has male and females as separate individuals. So they always have to outcross, and the same thing with water hemp. So those could be candidates for control with a gene drive system. Morning glory, on the other hand, has quite a bit of inbreeding. Some people think of it as a nice ornamental, but it's a major weed in soybean. That would probably not be a good candidate. Moving on to other parts of this figure, this idea of non-toxic pesticides and herbicides, I think this needs to be looked at much more carefully because the whole idea is to engineer a population so that it becomes susceptible to something that's not toxic. But I don't know if it seems to me that's been leapfrogged by the approach of using double-stranded RNAs, where you have something that's really a non-toxic, if it's done well, would be non-toxic and very specific. So it seems like you would be doing a two-step process using the gene drive instead of a one-step process. So it seems like the gene drives are better to have them working in a one-step process that is just limiting the population. And I just want to move back to the vector control thing. I know that Zach is going to deal in detail with mosquitoes, but I just want to bring up one other program to point out some of the social issues briefly, and that's the tsetse fly, which is a major problem in a large region in Africa because of trypanosomiasis, which there are different effects on human sleeping disease, but also for cattle. And this is the area in Africa that has trypanosomiasis, and this just gives you the number of different tsetse fly species that are in these different areas. But you also probably could see this is the area, one area of the most highest biodiversity in the world. So you have to think about what happens if you eliminate the tsetse fly. It could be wonderful. There have been some ex-ante economic studies that show that this would enable people to have better cattle and more productive dairies as well as beef production. But it just seems like we have this responsibility that if we're going to change a system like that, we have to think about what's going to happen to the people in reality. All right, so if you're going to do that, you have to think about which farmers are going to profit from this, who's going to lose their land, how is that going to go. So I think the issues in terms of you could do this, and people are definitely working with sterile tsetse flies and have had some elimination of tsetse fly, but if you're dealing with gene drive and you're going to eliminate it throughout an area like this, you have to be concerned about poverty and you have to be concerned about biodiversity. So that's what I have here. I just give you a summary here that, you know, basically gene drive technology has the potential to address major, really recalcitrant environmental and health problems. Like I mentioned with the Hawaiian honey creepers in the beginning, there's nothing else that seems to be able to address it. As I pointed out with the roaches and such, sometimes we don't have the tools yet to deal with those species, but it may be coming. It's critical to understand the population genetics and ecology, as I pointed out quite a few times, and to understand the social context. I would also say that one of the things that's typically, you know, keep coming up is that some of the easiest to develop, or what seem to be the easiest to develop, gene drive systems are non-self-limiting, so that if you release it, it's like Ice 9, it just goes and never stops. I think that the real challenge is to develop these spatially self-limiting gene drive systems. Okay, well, I'll stop there. I think I've used my time.
Thank you so very much, Fred. It's an excellent talk. So members of the committee, we have a few minutes to ask Fred some questions for the next 10 minutes. You will need to unmute yourself in order to ask those questions. For those joined by computer, there is a little red microphone tab. If it is red, you are muted. If it's gray or disappears, you are unmuted. For those on the phone, you will need to press #6 to unmute yourself, or *6 to unmute yourself.
This is Jason Delborn. A question for Fred. Thank you so much for a really nice overview. I wonder if you could expand a little bit on the idea that gene drive systems be spatially self-limiting and what that would accomplish if we could do that. Also, what are the likely strategies?
Yeah, thanks. Well, I really think this is an important issue. As we're moving into this area of gene drives, you certainly have a lot of social issues and what I would consider transboundary issues. So if, for example, you want to release something in one country and that country wants your gene drive, what happens if the bordering country doesn't want it? How do you limit those kind of things? So that it's clearly an important issue to have that kind of limitation, at least in the beginning. Certainly you can work with all those countries to come up with some harmonized approach so that everybody's okay with it, and I think that could be great in some cases. But in other situations, if you could limit the gene drive to a specific city where you want to release it, and you have approval, that would seem to be a step-by-step approach as opposed to just going full bore. In terms of systems that currently are available that would be self-limiting, I think they're much more likely in cases where what you're trying to do is take an organism that vectors a disease and making it incapable of vectoring a disease. So the kinds of things that are available in terms of self-limiting drives would have to do mostly with cases where there's underdominance, where the homozygote wild type and the homozygote that has the trait are have higher fitness than the intermediate heterozygote state. And under those kind of conditions, you have to have a critical threshold. You have to release something like 25 or 50% individuals into the population. So they start off at a gene frequency of about 25% or 50% in order for them to increase. So if a few individuals leave the city, they're unlikely to get up to that high proportion in other areas. This becomes more difficult when you're talking about population suppression, but there are things that could be done to cause those to not spread as well.
This is Lisa Tannehill. Can you hear me?
Yes.
Okay, great. Thanks again, Fred, for your presentation. I wanted to ask a clarification question with regards to gene drive usage in asexual species. You had mentioned that you can't use that type of gene, or you can't use gene drives effectively. And I was wondering if you could just clarify whether it's even possible to use gene drives in an asexual species, if you think there might be technologies on the horizon that would allow that to happen?
No, I don't think, as far as everything I know, there's no way to do that with an asexual species, because it all depends on your released individuals breeding with the general population. So there's no technology that I can think of that would get past that barrier.
Okay, thank you.
Any additional questions for Dr. Gould?
Fred, this is Joe Travis. So you're really, part of what you're pointing out is that in situations like the coffee borer, where you have presumably high levels of inbreeding, essentially it defeats outcrossing, or in morning glories that are self-fertilizing, you're really defeating outcrossing, and you're essentially struggling with something that is kind of like an asexual reproductive system, only very, although, sort of much, a little bit different, but essentially very, very low rates of outcrossing, very, very low rates of spread of the drive. That's what you're getting at with those examples?
Yes, exactly. So there's a continuum. It's not that you couldn't get anywhere with something that's partially inbreeding, but when something is partially inbreeding, you're probably selecting for them to become even more inbred.
Right. That's a really good point. Thank you. Wonderful presentation, Fred. Thank you.
You're welcome.
This is Fran Shovels. I have a question. Could you go back, please, to the chytrid fungus? You said that you couldn't engineer the fungus and you'd have to work with the frogs. Can you elaborate on that? I don't exactly understand why that would be the case.
Okay, where did I? Sorry, let's find this here. All right. So with the chytrid fungus, from I'm not a fungus person, but from what I have heard from people who do work with that, you wouldn't be able to have gene drive work because, again, about... Hello? Can you hear me?
We can hear you. Go ahead.
Okay, all right. Yeah. So it would be somewhat the same kind of a thing in terms of spreading those chytrid fungi throughout the environment, as well as their own mating system. So as I said, I'm not the expert. From what I've heard from experts, that would not be something that you could do, is engineer that fungus and expect it to spread.